In this video, we will learn about the process of digestion. Digestion is a process of physical and chemical breakdown of large and complex molecules of food into simple and small molecules by action of digestive enzymes so that they can diffuse into the blood through the gut wall. First is the digestion in buccal cavity. The ingested food is chewed and masticated in the buccal cavity with the help of teeth. Mucus in saliva moistens and lubricates the food, making it soft so that it can be swallowed easily. Salivary amylase present in the saliva hydrolyzes starch into maltose. Next is pharynx and esophagus. No mechanical or chemical digestion occurs in pharynx and esophagus. It only passes the food from the buccal cavity to the stomach with the help of peristaltic movement. Next is digestion in stomach. Protein digestion begins in stomach. The muscular wall of stomach undergoes alternate contractions and relaxations and churns the food into fine pulpy mass called chyme. Gastric juice contains hydrochloric acid. Digestive enzymes such as pepsin, renin, and gastric lipase, and mucus, that is gastric mucin. Now let's understand each of these gastric secretions one by one. First is hydrochloric acid. It makes pH of food from alkaline to acidic, that is below pH 7. It destroys bacteria present in the food and activates the inactive propepsin into active pepsin. It also controls opening and closing of the pyloric aperture. Now let's understand about the enzyme secreted in stomach. First enzyme is pepsin. It is secreted in an inactive form pepsinogen, and in the presence of hydrochloric acid it is converted into active form pepsin. It hydrolyzes the protein molecules into proteoses and peptones. Here we have the reactions. Pepsinogen, that is inactive form, is converted into pepsin, that is active form, in the presence of hydrochloric acid. Proteins are converted into peptones and proteoses by this enzyme pepsin. The next enzyme is renin. In presence of hydrochloric acid and calcium ions, renin help in the digestion of milk protein. Renin is present in inactive form, that is prorenin, which is converted into the active form renin in the presence of hydrochloric acid. Renin along with milk protein casein forms paracasein, and this paracasein along with calcium ions form calcium paracaseinate, which is insoluble. Now this calcium paracaseinate is acted upon by pepsin and it is converted into proteoses, peptones and polypeptides. Next enzyme is gastric lipase. It partially hydrolyzes fats into fatty acids and glycerol. Next is gastric mucin. It protects the mucous membrane of stomach against the action of hydrochloric acid. Now let's understand about digestion in small intestine. First is digestion in duodenum. The partially digested food, that is chyme from the pyloric stomach, is pushed into the duodenum by relaxation of pyloric sphincter. The bile juice from gallbladder and pancreatic juice from pancreas are mixed with chyme in duodenum. Now let's understand functions of bile juice. Bile juice has no digestive enzyme, but it is important for digestion because first, bile neutralizes acidity of chyme in the duodenum. Second, it makes medium alkaline for the action of enzymes of pancreatic and intestinal juice. Third, bile emulsifies fats and prepares them for the action of enzymes. Now let's understand the role of pancreatic juice. Pancreatic juice contains following enzymes. First is trypsin and chymotrypsin. Both trypsin and chymotrypsin are secreted in an inactive form called trypsinogen and chymotrypsinogen. On being changed to active form, they bring about protein digestion into polypeptides. Second is carboxypeptidase. It hydrolyzes polypeptide chains into tripeptides, dipeptides, and amino acids. Third is amylopsin or pancreatic amylase. It hydrolyzes the remaining starch and glycogen into maltose, which is a disaccharide. Fourth is tiapsin or pancreatic lipase. It acts on emulsified fats and hydrolyzes them into fatty acids and glycerol. Fifth is nuclease and nucleotidase like ribonuclease and deoxyribonuclease. These hydrolyze nucleic acids, that is DNA and RNA, into nucleotides and nucleosides. Next is digestion in ileum. Ileum is the second part of small intestine. Small intestine is the site of complete digestion of food, that is carbohydrates, proteins, and fats. The Brunner's glands and the crypts of lubricant secrete intestinal juice called succus entericus, and it contains following enzymes. First is erepsin or peptidase. It hydrolyzes polypeptides, tripeptides, and dipeptides into amino acid. Second is maltase. Maltase hydrolyzes maltose, which is a disaccharide, into glucose, which is a monosaccharide. Third is sucrase, which hydrolyzes sucrose into monosaccharides. Fourth is lactase, which hydrolyzes lactose into glucose and galactose. Fifth is lipase, which emulsifies the remaining fat into fatty acids and glycerol. Sixth is enterokinase, which converts inactive trypsinogen into active trypsin enzyme. Succus entericus also contains nuclease, nucleotidase, and nucleosidase enzymes which hydrolyze nucleic acids, nucleotides, and nucleosides respectively. Next is digestion in large intestine. Water is absorbed by the mucous lining of large intestine and undigested residue is changed into semi-solid feces. These are pushed into rectum from where they are passed out through anus. This is called as egestion or defecation. Here we have the large intestine where the absorption of water takes place, and this part is called as rectum where the feces are temporarily stored and then it is defecated or egested out of this anus. I hope this topic is clear to you. Thank you.